Somewhere inside the nucleus of nearly every single cell in your body, tucked quietly among the genes responsible for building and running the human brain, sits a small stretch of genetic code that did not originally belong to humans at all. It came from a virus. Not a virus currently infecting you and not one your immune system is actively fighting off right now, but an ancient viral invader that infected one of your extremely distant ancestors. Millions of years ago, inserted its own genetic material directly into that ancestor's DNA, and then, remarkably, never left. According to a growing body of neuroscience research, that ancient viral remnant did not simply sit there harmlessly for millions of years afterward. It appears to have been quietly repurposed by the human brain itself, and it may be doing something genuinely essential for how memories are formed at this exact moment inside your own head, as you listen to this sentence. For a very long time, the scientific and medical community referred to this category of genetic material using a term that reveals exactly how little value researchers originally assigned to it. They called it junk DNA. For decades, roughly 8% of the entire human genome, an enormous stretch of genetic material, was believed to consist largely of inactive, leftover viral fragments, ancient evolutionary debris left behind from countless infections that occurred across millions of years of primate evolution, fragments assumed to serve no meaningful biological function whatsoever inside a modern human body. That assumption, it turns out, was significantly premature, and in at least one genuinely remarkable case, it was almost completely wrong. To understand how a virus ends up permanently embedded inside human DNA in the first place, it helps to understand a very specific category of virus known as a retrovirus. Unlike most viruses, which simply hijack a host cell's machinery temporarily in order to reproduce before eventually moving on, retroviruses do something considerably more invasive and considerably more permanent. They insert a copy of their own genetic material directly into the host's own DNA, embedding themselves within the host genome itself. If that infection happens to permanent, inheritable part of that species' genome moving forward, generation after generation, indefinitely. Over the vast span of primate evolutionary history, this exact process happened repeatedly, again and again, embedding countless ancient retroviral sequences permanently throughout the human genome. Most of these ancient viral remnants, sometimes called endogenous retroviruses, did eventually become genuinely inactive over time, accumulating mutations across millions of years that left them incapable of causing any further infection or producing any functional protein whatsoever. But not every single one of them stayed silent. A small number of these ancient viral genes appear to have been gradually captured, repurposed, and integrated directly into essential biological processes taking place inside the human body, including, in one particularly striking case, inside the human brain itself. Researchers estimate that endogenous retroviral sequences collectively make up somewhere around 8% of the entire human genome, a considerably larger proportion than most people would ever assume, given how rarely this fact makes its way into ordinary science education. For comparison, the genes that actually code for functional human proteins, the genes most of us picture when we think about DNA in the first place, make up a notably smaller percentage of the total genome than these ancient viral remnants do. For decades, this striking imbalance was simply treated as further evidence supporting the broader junk DNA assumption, the idea being that if something occupied that much space inside the genome without producing any obvious functional protein, it most likely served no meaningful biological purpose at all. Advances in genetic sequencing technology and increasingly sophisticated laboratory techniques have gradually chipped away at that assumption, revealing that a meaningful portion of this supposedly inert genetic material is, in fact, actively involved in regulating when and how nearby genes are switched on or off, even in cases where the ancient viral sequence itself no longer produces a functional protein of its own. If you find it fascinating that ancient viral infections quietly became a permanent part of human biology, go ahead and hit that like button right now. It genuinely helps this channel keep bringing you deep dives, exactly like this one. The gene at the center of this particular story is called ARC, short for activity-regulated cytoskeleton-associated protein. Neuroscientists have known for decades that ARC plays an absolutely essential role inside the human brain, specifically in a process called synaptic plasticity, the biological mechanism responsible for strengthening or weakening the connections between individual neurons based on experience, which is the fundamental process underlying how memories are formed and stored in the first place. Without properly functioning ARC, protein laboratory research has consistently shown that the brain struggles significantly to convert short-term experiences into stable, lasting long-term memories. What researchers did not fully appreciate until considerably more recently is exactly how unusual the ARC protein genuinely is compared with virtually every other gene found anywhere throughout the rest of the human genome. detailed structural and genetic analysis eventually revealed that the ARC gene shares a strikingly close resemblance to genes found inside retroviruses, the exact category of ancient viral genetic material capable of permanently embedding itself within a host's DNA. Researchers studying this connection eventually determined that the ARC gene most likely originated from an ancient retrovirus that infected a distant ancestor of modern mammals somewhere around 350 million years ago, long before humans, or even primates more broadly, existed anywhere on Earth. Here's where the story becomes genuinely difficult to fully wrap your head around. Researchers studying the ARC protein directly discovered that it does not simply resemble a viral gene in a distant, abstract evolutionary sense. It actually behaves like one in a very direct and observable physical way. When researchers examined ARC protein under close laboratory observation, they discovered it spontaneously assembles itself into small, hollow, capsule-shaped structures that are, in terms of overall physical shape and structure, virtually indistinguishable from the protein shells that actual retroviruses use to package and transport their own genetic material from one infected cell to another. Even more remarkably, researchers discovered that these ARC protein capsules do not simply sit motionless inside brain cells, performing some kind of passive structural function. They actively package up genetic material, specifically a molecule called messenger RNA, encase that genetic material inside their small capsule-shaped shell, and then get released directly outside of the neuron itself, where they subsequently get taken up and absorbed by a neighboring neuron located nearby. In other words, brain cells appear to be using this repurposed ancient viral machinery to send genetic material back and forth directly between neurons, a genuinely unusual form of intercellular communication, unlike virtually anything else observed occurring naturally elsewhere throughout the human body. The research team that first documented this behavior, working independently across 2 separate laboratories, studying fruit flies and laboratory mice, published their findings within the same scientific journal issue. A genuinely notable coincidence that only strengthened confidence in the overall discovery. Both research groups, working with entirely different animal models and using different experimental techniques, arrived at strikingly similar conclusions regarding how ARC protein behaves, how it assembles itself into these virus-like capsule structures, and how it appears to transport genetic materia L between neurons. This kind of independent convergence across separate laboratories is exactly the kind of scientific confirmation that transforms an interesting, isolated observation into a genuinely credible and widely accepted biological finding. To put this discovery into perspective, imagine 2 neurons sitting beside one another inside your brain, separated by a microscopic gap called a synapse, the same basic structure responsible for essentially all communication happening between neurons throughout the entire nervous system. Ordinarily, communication across that gap happens through chemical signals called neurotransmitters, molecules released by one neuron that bind to specific receptors on the neighboring neuron, triggering an electrical response. What researchers discovered with ARC protein represents an entirely different additional layer of communication happening alongside that more familiar chemical signaling process, one that involves directly transporting genetic instructions from one neuron into another, potentially influencing which proteins that neighboring neuron goes on to produce, and ultimately shaping how strongly or weakly the particular neural connection continues to function moving forward. If uncovering these deep, unexpected connections between ancient viruses and modern human biology fascinates you, make sure you are subscribed, because there are countless more incredible discoveries just like this one still waiting to be explored right here on this channel. Researchers studying this phenomenon believe this viral-like transport mechanism may play a genuinely important role in how neurons communicate with one another during the formation of new memories, essentially allowing one neuron to directly influence gene expression happening inside a neighboring neuron by physically transferring genetic instructions between them using a delivery mechanism originally evolved by a virus for an entirely different purpose millions of years ago, rather than the ancient retrovirus infecting and harming its host. In this specific case, the host appears to have successfully captured the virus's own genetic material and delivery machinery, and repurposed the entire system for its own essential biological benefit instead. This process, in which a host organism captures viral genetic material and repurposes it for its own biological advantage, is not entirely unique to the ARC gene alone, though the ARC discovery remains one of the most striking and thoroughly studied examples found so far. Researchers studying the broader human genome have identified several other examples of ancient viral genes that appear to have been similarly repurposed for legitimate biological function over the course of evolutionary history. One particularly well-known example involves a gene called syncytin, originally derived from an ancient retroviral infection, which now plays an absolutely essential role in the formation of the human placenta during pregnancy, allowing cells to fuse together in a manner directly borrowed from the exact same mechanism ancient retroviruses originally used to invade and fuse with host cells during active infection. Without a properly functioning syncytin gene, the placenta cannot form the specialized fused cell layer necessary to allow nutrients and oxygen to pass efficiently from a pregnant person's bloodstream into the developing fetus, meaning this ancient viral gene has become quite literally essential for successful human reproduction itself. Researchers have found similar syncytin-derived genes performing comparable placental functions across a wide range of other mammal species as well, though, remarkably, in several cases, different mammalian lineages appear to have independently captured and repurposed entirely different ancient retroviruses for this exact same reproductive purpose. A phenomenon evolutionary biologists describe as convergent domestication, where evolution arrives at a similar biological solution multiple separate times using different genetic raw material each time. Taken together, the ARC gene and the syncytin gene represent two of the clearest, most well-documented examples of what researchers call viral domestication, a process through which genetic material that originally served a virus's own reproductive interests gets gradually captured, modified, and integrated into the host organism's own biology, eventually serving a purpose entirely disconnected from, and in some cases directly opposite to, the original viral infection's purpose. Both examples suggest that this kind of viral domestication may have played a considerably larger role in shaping essential aspects of mammalian biology, including both reproduction and cognition, than researchers ever seriously considered before these specific discoveries were made. Naturally, a discovery this significant did not arrive without a considerable amount of careful scientific scrutiny, and that scrutiny is genuinely worth understanding rather than dismissing outright. Some researchers have raised legitimate and important questions about exactly how universally this viral-derived ARC mechanism actually operates across different species, different brain regions, and different specific types of memory formation. Much of the most detailed experimental evidence supporting this theory has come from studies conducted using fruit flies and laboratory mice rather than direct experimentation on human brain tissue, for obvious and important ethical reasons involving human research limitations. So what does this genuinely mean for our broader understanding of human biology and human evolution? For one, it means the sharp, clean boundary many of us likely assumed existed between virus and host, between foreign invader and legitimate biological self, may actually be considerably blurrier than most of us ever previously imagined. An organism's genome is not simply a fixed, static blueprint passed down unchanged from one generation to the next. It is a considerably more dynamic, considerably more layered historical record, one quietly shaped across millions of years by countless ancient infections, many of which left behind genetic material that evolution eventually found a genuinely useful new purpose for, long after the original threat those infections once posed had completely disappeared. It also means that some of the most fundamental biological processes taking place inside your own brain right now, at this exact moment, as you form new memories simply by listening to and processing this sentence, may ultimately trace their origin back to an ancient viral infection that occurred hundreds of millions of years before humans, primates, or even most modern mammals existed anywhere on the planet. That is a genuinely strange thing to sit with directly. The same broad category of biological process, responsible for viruses, spreading disease, and causing illness throughout human history, was also, through an entirely separate and unrelated evolutionary pathway, quietly captured and repurposed into becoming one of the essential biological tools your own brain currently relies upon in order to learn, adapt, and remember. This raises a genuinely fascinating broader question worth sitting with. If a single ancient retroviral gene was successfully captured and repurposed for such an essential biological function, how many other genuinely important pieces of human biology, still sitting quietly unexamined somewhere throughout that previously dismissed junk DNA, might similarly turn out to serve genuinely essential biological functions researchers simply have not yet discovered or fully understood? The Human Genome Project, completed in the early 2000s, provided researchers with a complete map of human genetic material, but having a complete map is not remotely the same thing as fully understanding what every single piece of that map actually does. Scientists are still, even now, many years later, continuing to systematically work through that enormous genetic landscape, discovering that considerably more of it serves a genuine, meaningful biological purpose than researchers originally ever assumed. This gradual shift in scientific understanding mirrors a broader pattern that has repeated itself many times throughout the history of biology. Structures and genetic sequences once dismissed as vestigial, purposeless, or simply left over from earlier evolutionary Stages have, again and again, eventually revealed themselves to serve genuinely important functions once researchers developed sufficiently precise tools to study them properly. The label junk DNA itself, still occasionally used in casual conversation even today, is increasingly viewed within the scientific community as more of a historical artifact reflecting the limitations of earlier research technology than an accurate, lasting description of what that genetic material actually does inside a living human body. There is something genuinely humbling in sitting with this discovery for a moment. Every single one of us alive today carries within our own DNA a quiet, permanent record of countless ancient viral infections, stretching back across hundreds of millions of years of evolutionary history, long before humans, primates, or even most modern mammals existed anywhere on the planet. Most of that ancient viral material genuinely is inactive, harmless leftover debris, exactly as researchers long assumed, but at least one small, specific piece of it appears to have been quietly captured, repurposed, and transformed into something genuinely essential, a tool your own brain is very likely actively using at this exact moment, helping you form and store the very memory of watching this particular video. So when a headline tells you that scientists discovered a virus hidden inside human DNA that has been silently shaping the brain, understand exactly what that claim genuinely represents, and just as importantly, what it carefully does not mean. It does not mean there is some active, ongoing viral infection currently occurring anywhere inside your brain right now, and it does not mean this ancient genetic material poses any kind of active health threat or danger whatsoever. It means that millions of years ago, a virus permanently inserted a small piece of its own genetic material into the DNA of one of our extremely distant ancestors. And that evolution, working patiently across an almost unimaginable span of time, eventually found a genuinely essential new purpose for that ancient viral remnant, repurposing it into becoming one of the fundamental molecular tools the modern human brain now relies upon in order to learn and remember. Perhaps that is the real lesson worth carrying away from this particular story. Evolution rarely, if ever, builds entirely new biological tools completely from scratch. Far more often, it works patiently with whatever genetic material already happens to be available, however unlikely or strange that original source material might initially seem, gradually reshaping, repurposing, and refining it across almost unimaginable stretches of time until something entirely new, entirely unexpected, and, in this particular case, entirely essential to how you and every other human being alive today are able to think, learn, and remember, quietly emerges from what was once, however briefly, nothing more than an ordinary and ancient viral infection. And somewhere inside your own brain, right at this exact moment, that ancient viral remnant is very likely already at work, quietly folding itself into small capsule-shaped structures, packaging up genetic material, and passing it along between neurons, helping turn this very sentence into a memory you may still be able to recall days, months, or even years from now. 350 million years separate you from the ancestor first infected by that original virus. And yet, somehow, a small fragment of that ancient infection never actually left. It simply waited patiently until the human brain finally found a use for it.